Good morning, ladies and gentlemen. Thank you for being here with us for today's online event on Pandemic Proofing Society, partnered with the National University Health System and A-STAR's Bioinformatics Institute. My name is Alethea from the National Supercomputing Centre Singapore, or NSCC for short. For those of you who are not familiar with us, NSCC is a national research infrastructure funded by the National Research Foundation of Singapore. NSCC is Singapore's first petascale supercomputer facility that provides high-performance computing and resources to all local research institutes, institutes of higher learning, government agencies, and companies in both the public and private sector. HPC resources are beneficial in a variety of ways in a number of different sectors. It enables research of the highest computational complexity and data intensiveness in areas like advanced modeling and simulation, artificial intelligence, and big data analytics to be completed in shorter time spans. If you are interested, do find out more about NSCC and HPC at nscc.sg. Before we get started with the talk, please do note that if you have any questions, do post them into the Q&A chat at the bottom of your screen. We will address your questions during our Q&A and panel discussion segment at the end of the presentations. During the panel discussion segment, you may also choose to raise your hand, and we will bring you on screen to speak. So without further ado, let me introduce our moderator, Dr. Kenneth Ban, who will be facilitating today's session. Dr. Ban has been a medical educator in the Department of Biochemistry, Faculty of Medicine, National University of Singapore since 2011. He graduated from NUS Medicine and completed his graduate and postdoctoral studies in cancer biology and mouse genetics before joining NUS Medicine. He is currently the Program Director for Phase 1 Biochemistry and is actively involved in writing collaborative case learning cases to help students understand how basic science concepts can be applied to clinical cases. So let me now give the time over to Dr. Ban. Dr. Ban, please.
Hi, good morning, everybody. Pleasure to have all of you here to hear talks by two eminent speakers. I will touch upon how digital transformation is affecting health and biomedical sciences. So I'd like to introduce our first speaker, Associate Professor Ngiam Yuan. He's the Senior Consultant Division of Thyroid Endocrine Surgery at the National University Hospital. He has many roles. Among them, he's the Group Chief Technology Officer in the National University Healthcare System. He oversees technology deployment in the Western Healthcare Cluster of Singapore. So he assists the Chief Executive in implementing new technologies. So he is concurrently also the Deputy Chief Medical Information Officer at UHS, and his specialty focus is on artificial intelligence research and information technology implementation in healthcare. So without further ado, Professor Ngiam.
Thank you, Kenneth, for the kind introduction. Yeah, it's quite formal, but we just had a number of interactions in the last week. So I thank you for introducing myself. Okay, so very quickly today, I'm going to talk a bit about how digital medicine, in particular precision medicine and live data, can change the way we function in the post-pandemic world. Of course, we are now in the phase of the pandemic where, at least in Singapore, we are seeing a lot, very minimal community transmission. And it gives us time to pause and reflect and think about how we can do better, as well as what the future looks like in terms of precision medicine. And some people might wonder what's precision medicine got to do with the pandemic. Well, as you know, it is pretty much everything, right? Because the pandemic has brought to light the importance of investment in science, as well as in tools that allow us to quickly detect, trace, as well as to treat pandemic-related issues. So this touches on the point of a mindset of digital transformation, which is really about saying that, okay, we have these amazing digital tools in hand, and we want to use these tools better to improve our transform the way we transact medicine, as well as to look at how research can be integrated into our daily activities when we talk about bench to bedside. So other than just providing better services at lower costs and improve efficiencies, right? We hope that the research element can also be integrated into our day-to-day work, especially in the time of pandemic. So, you know, for those who have been working in this field, right, we will find that the inertia to actually move towards a new fields is actually really, really high, because there are a lot of established and legacy systems that do not allow us to move things forward. And when it comes to precision medicine, right, this is a relatively new field in actual production medicine, because it's safe to say that the current healthcare systems are not designed to support precision medicine in the way we think it is, specifically things like genomics, as well as running things like COVID-19 tests and upcoming vaccination kind of research. So we need to invest large amounts of not just money but effort into ensuring that these lab innovations can be translated into actual care. And in my area of work, it translates to data, right? Having the data to be able to make those changes would be the key to allowing us to translate what we find in the lab into an electronic form, with which then we can then use that data for the purposes of discovery and research, right? So therefore, there's an adage where people say that data is the new oil, and it's very true, right? Because the tools that we use in today, such as the neural networks and the AI tools that we use today, all depend on having big data, and having large data sets would allow us to build not just multiple different kinds of models, but highly accurate models for the purposes of addressing specific needs. Like, for example, how would you then do contact tracing? That's the kind of information that if you can pool data together, that would allow us to then be more effective in tracing and more effective in modeling how the pandemic is being spread. Okay, I think this is the current state of thought when it comes to looking at data at rest and data in a certain place and running models on particular data. But I want to introduce a slightly different take on this, which is to say that, you know, being able to build models does not mean that you can actually deploy anything. Our experience in running and building and analyzing and running big data sets didn't actually result in immediate deployability. And the reason is because we don't have the key factor, which is having live data. Having real-time live data is a game changer. And I'll come to that in a minute to explain why that's the case. But before you go there, right, if you look at precision medicine, right, and if you see how, what are the factors that affect individual health outcomes, right, you can see that the vast majority of health outcomes are 40% lifestyle factors, right? And again, to touch on the pandemic, right, a lot of the vectors or the way pandemic is spread is because of the way we undertake our lifestyles, right? And in this post-pandemic world, a lot of lifestyle changes needed to be made to prevent the spread of the disease, right? And there are certainly certain groups of people who are more susceptible to the disease than just than others. And this is partly result of aging, but we also know that there's a genomic component to it, although it's less clear who might be more susceptible from a genetic basis. Social environmental factors are probably more of chronic condition, chronic disease kind of factor. And of course, 10% of it is healthcare. So we're quite blessed in Singapore to not have had our healthcare system overwhelmed by the people who are sick from coronavirus. However, that said, it could very well be the case where if we weren't able to modify our lifestyle factors, that is, you know, go into lockdown, stop people from going out, from mingling, we may actually be taxing that 10% of healthcare that affects outcomes. So just a quick point to this, which is precision medicine really takes into account, you know, all factors, right? Genes, environment, and lifestyle of each person. And On a more long-term chronic basis, unrelated to the pandemic, which is we can use that to predict risks, to tailor therapies, and to make more accurate diagnosis, and also to reduce adverse events to reactions that to our treatments that we give our patients. And this is premised on building the analytical tools and models that allowed us to make those predictions. So just to have a high-level overview of where we are in terms of precision medicine, right? And how is it relevant? Well, we are right now in phase 2, where we are increasing the number of population that we would like to sequence to about 100 to 150,000. And we certainly want to look at build disease cohorts. And here I would like to add disease cohorts that involve, for example, COVID patients. And also look at new and different kinds of data types that are relevant to the condition. And in the next 2 to 3 years, we will probably be achieving these numbers and generating large amounts of data that will allow us to process and perhaps come up with very specific insights related to precision medicine. In particular, I want to mention the field of pharmacogenomics, because pharmacogenomics is one of those fields that avail themselves to sort of early wins, because everybody takes certain kinds of drugs. We do know that certain genes have an impact on the efficacy as well as the rate of side effects of certain drugs. So certainly in terms of the bang for the buck, as it were, I think drug pharmacogenomics is one of the key areas that we are looking at in terms of precision medicine initiatives. And of course, beyond 2023, we really want to look at integrating newer sources of data, things like lifestyle and environment data that are actually quite hard to capture on an individual basis, and also to ensure that the precision medicine workforce is trained to allow us to deploy all these technologies that are out there. Okay, so for those of you who are not privy to the journey of precision medicine, actually precision medicine is really quite an arduous journey, right? It's not as simple a thing as just taking somebody's blood or tissue samples and running it through a sequencer and getting results from a sequencer, right? So there are issues with access to testing, cost, privacy issues, real or perceived lack of utility of the tests, choosing the right tests, ensuring that the patients who use these tests are counseled, and so that they understand what the implications of having a positive test, taking the right consent from the patient, they are HPRA compliant, collecting samples. Something as simple as transfer to the lab might seem very rudimentary, but actually it is something quite difficult, because on top of the usual requirements for production-ready labs, we need to automate some processes to be able to pick up the patient's samples that have the appropriate consent. So this consent needs to be electronic and automated. The testing itself requires a certain amount of regulatory approval as well as manpower training. Here we're talking about getting the right analysis for variant calling and so on and so forth. Data interpretation, storage of data, as well as how do you then automate the return of the data to the physicians. And finally, with the results, how can we do appropriate genetic counseling, as you can see in the pre-test, and now is the post-test genetic counseling, and follow-up and the management of the patient. So the ultimate goal really, of course, is to help our patients have better results that was previously not possible. But there are other things that that come from achieving that aim, which is to have a vibrant health and biomedical sciences sector that's optimized for value creation and capture, as well as a data-driven healthcare systems for population needs and optimizing treatment and prevention. So as you can see, all these are premised upon having the data and having data at the right place to the right people to run the appropriate modeling and analysis. So in NUHS, we are quite clear about this, right? We are not just doing this for the purposes of like following everybody else in doing this particular precision medicine initiative. We are committed to using AI tools to help us support this journey. And it's, when I say AI, really in a clinical practice setting, it's about augmented intelligence. It's not about replacing the doctor. It's really about providing the doctor with insights that he or she would previously not be able to access and use that information effectively to treat patients. So, you know, AI is really our healthcare's fourth industrial revolution. Most of you have heard about Industry 4.0. In healthcare, we're a bit behind, and certainly we are really talking about going from electronic medical records that are analysis intensive to the third generation, which is analyzed insights, and meaning that the tools, the AI tools, will actually give us the precision medicine insights that will help us process all that large amount of information and to give us insights that permit us to use our experience to treat the patient appropriately. So it's not just a fully automated process where the machine is doing all the thinking. It is really about a combination of the machine and the clinician to deliver the right kind of care to our patients. So to develop these processes, it's really data heavy, but it really comes down to the clinical use case, right? So in this particular scenario, if you pick one scenario where we are using pharmacogenomics, so pharmacogenomics is the problem definition, meaning that can we use excuse me, pharmacogenomics to support the appropriate treatment of our patients with the correct drug dosing to reduce the impact of side effects as much as possible. So that might be a problem statement. Then the next phase is really about extracting and refining the data. And we spend most of our time here preparing the data. And indeed in pharmacogenomics, this is really the key bugbear, right? Which is the ability to sequence the data and store it and do variant callings to allow us to then present that data with linkage to the phenotypes, and finally to be able to then suggest appropriate treatments for our patients. So these are where the analytical and model validation comes in. Here there's an element of human-machine interface where doctors need to know how to interpret this information. So a level of training is required, as well as the need for these interfaces to be tailored to the doctor's care processes. So for example, we require some form of clinical integration with the EMR system, so the clinical deployment issues need to be addressed before we can actually see an effective solution to the problem. And here we are just listing some of the other domains that that we have been working on. So augmenting the doctor, which is exactly what I just mentioned earlier. So using pharmacogenomic insights, precision medicine insights to augment doctors' treatment of patients, improving the safety, so predicting risk of falls and etc. So this is more of an AI piece. Doing drug discovery applications. So this is even more so, ever more so important in this year and in many years from now. I think most of us have heard some about the breakthrough in protein folding prediction from AlphaFold. So this is the kind of work that we hope to do that allows us to use computational methods to predict certain fundamental scientific issues that might have downstream translation capabilities. The rest are some of the process and finance related issues, as well as some behavioral issues that we can address with AI in the community. So last year we published a short article, a review of how we can bring big data and machine learning algorithms into healthcare delivery. And this is where we emphasize the key point, which is you can spend a lot of time collecting data. And what I'm showing here is our current platform, which is Discovery AI, which we use to collect and link, de-identify and link data. as well as to automate the process of building AI tools to be able to give insights to our doctors. So just very quickly to take you through this rather noisy diagram. So we actually get data from our EMR system, which is going to be epic in the coming year. And that information is pushed into our virtual machine, our master virtual machine, which is then processed and fed into our AI tools here on the right. So these AI tools then process that live information and push out the suggestions to the doctors via our EMR interface. So this is what I'm talking about, building the correct EMR interfaces to augment clinicians' workflows. But to build all these AI tools, you need a place to store all the different data sets as well as to link them up in an effective way. So what we do is that we actually provide virtual machines to our researchers to be able to run their projects on large data sets. And when they build their data set, we can completely onboard this new data on a daily basis to these models, which will then start to output suggestions to doctors. So this is really a sort of an AI foundry to build new AI tools that will support our clinical workflows. So I just want to say that, you know, in the years that we've been trying to do this, not just for precision medicine, for any kind of AI tool building, right? Real data is the key thing, and I call it liquid gold. Why? Because with real data, you can truly realize the benefit of building AI models and building rule sets that allow you to take this real life data and transform it into insight. And you couple it with the ability to integrate multiple streams of data. That is, you're able to take different data sources from different places into a particular tool or machine to give you an insight that you can deliver to your clinicians. And this would be extremely powerful setup that will allow us to scale up on the kind of processes and as well as the different range of tools that we can potentially onboard onto our platforms. And transforming, in order to do this, right, one needs to think about the way we architect the underlying solution. You need to transform data at rest to data in motion. And this fundamentally changes the way we apply insights to data and actionable outcomes, right? And the architecture I'm really talking about is using messaging systems and microservices architecture, which is the current state of the art, right? The current modern architecture. So any of you who use Facebook or LinkedIn would be using messaging systems and event-based architecture to transact all these insights that we are seeing. So to do that, we've evolved what you saw earlier, which is a discovery AI platform, into what we call a tribrid. So we have actually 3 separate platforms that allow us to transact the different levels of AI development in the appropriate space, right? So here, to just talk you through this very simple, rather noisy diagram, here's right at the bottom here is what we saw earlier on Discovery AI. So the ability to aggregate, de-identify data and make it available to data scientists and clinicians to run their analysis as well as to build models is what happens here at Discovery. We are currently building and will hope to deploy the Endeavor AI, which is a production level system where all these models that we've built can be onboarded, and where live data coming from our EMR system will then be used to pass through these AI tools and give alerts back to the Epic BPA system. And finally, we also have a layer which is sitting on a commercial cloud. Now, this commercial cloud is there so that we allow industry to be able to collaborate with us. You can see here that we only have restricted data at this layer. That's due to regulations. However, this restricted data itself will actually permit many of our collaborators to be able to test out their innovations in a scalable commercial cloud space. And it also allows us to connect with our users on the internet. So just a very quick mention about messaging architecture and microservices, right? I think this is a kind of data streaming architecture that is required for you to execute model building and deploy it in an agile fashion, in a scalable agile fashion, as well as using reusable microservices. Just to maybe just mention this, right? So imagine here is Endeavour with its streaming data. So each of these columns are actually referring to streaming data fields. And what we can build here is our microservices that that anchor specific types of services like AI services, chatbot services, AI image services. And these are then used singly or in multiply by different service applications which consume different data, different views of streams of data to give an output to the users. So microservices here are reusable by different service applications consuming streaming messages to deliver a service to the user. So where are we now in terms of clinical grade genomics? Well, we are not quite there yet. My hope is that in the next 2 to 3 years, we will see widespread use of genomics. And this is largely driven by the cost, right? I mean, next generation sequencing costs have fallen by almost 10 times from the 2010 to 2019. And we hope that in the next 2 to 3 years, it will fall even more. And this will open the door to commercial genomic testing that allows us to deliver insights that will support our clinicians, advice to our patients, right? And certainly, we need to spend even more time ensuring that the workflow is sound, as well as to validate its effectiveness down the line. So I think in the post-pandemic world, having such a setup would certainly add to our agility and our ability to deliver really cutting-edge next precision medicine to our patients in a scalable way. And in NUHS, we are somewhere there. We are on our way. We are somewhere here right now, level 2. We are implementing our AI tools. We are building our pharmacogenomic pathways. We hope to go into the realm of multi-domain machine implementations throughout our cluster and eventually reach some level of, reach a high level of automation that is what they may term as artificial general intelligence. But I think we are many, many ways away from that. And there are some things to help us along the way. I mean, we do have the clinicians to solve our issues with us. We have so much data that I think that we don't even have enough people using it. We are very blessed to have our data scientists work with us. And again, with the support from partners like NSCC, we're very blessed to have scalable, secure hardware that we can run some of these analyses on. And we have the ability to run clinical trials and deploy on our platforms. With that, I would like to end here, and I'm happy to take your questions.
So thanks very much, Prof. Ngiam. So this was a very insightful talk and looking the future of how how digital transformation can affect healthcare. We will, just a reminder, we will take the questions at the end of the second talk so that we can consolidate everything. So please post the questions in the Q&A and then later we'll go through one by one. All right, thanks very much. So next, I'd like to introduce you to our next speaker. This is Dr. Sebastian Maustro. And Dr. Sebastian here is an expert in computational sequence and structure analysis in A*STAR Bioinformatics Institute. He's no stranger to many of us. He has worked on the National Precision Medicine Project and more recently has made critical contributions to national and global viral pathogen surveillance programs. So some tools that you may be familiar with, flu server and COVID server to interpret viral mutations, has been launched by him. He works closely with the global GSAID team to enable sharing of real-time analysis of pandemic coronavirus data. So he will be the new executive director for BII in coming January. So we also congratulate him for that. And without further ado, we look forward to listening for this talk on COVID-19.
Thank you very much, Ken. Thanks to all who listen, and also thanks to the previous speaker. Of course, it's very interesting, and precision medicine is also something where ASTA contributes, including from the structural bioinformatics side. And it's also very exciting to hear about the AlphaFold that opens up new opportunities. I'll be showing you today a slightly different application area that is also needing computation. And I will talk about the real-time surveillance of pathogens and how to use, for example, the COVID pandemic as an example, how computational work can help. As you know, when you start getting a sample and you do next generation sequencing, there is already a first computational step here to get all the reads either assembled or mapped to a reference sequence. So this is a first computational step. Then once you have the sequence, you want to make sense out of it. And I will also spend some time on that. But we shouldn't forget that in order for this to work effectively, for public health to be able to respond, timely sharing of those sequences is important. And this is something that is not so trivial, because people that would have those new outbreak sequences, they also have interest. They want to maybe patent, or they want to write the first paper. Interestingly, the writing the first paper seems to be one of their main driving factors, that they wouldn't share sequences freely. However, luckily, there are systems where you can do that and still get your first author paper in New England Journal of Medicine and Lancet and whatsoever. And this machinery is called GISAID. It's a global initiative that allows sharing of sequences where you keep the rights to your data, and others need to acknowledge you and need to ask you also what you want to do with it. But at the same time, everyone, especially public health colleagues, can look at these sequences. So you've been working with GISAID for over a decade, especially on influenza before. And when the new coronavirus came in, the outbreak started in China, the Chinese colleagues were contacting GISAID, asking if also the coronavirus genomes could be shared via GISAID. And in these very early days, I was one of the few who were trying to put up together a new system. How can we, it's a different virus, we actually needed to make a new database. And with very few people, a programmer in Germany, the president of GISAID is an IP lawyer who knows all about the intellectual property, as well as he has the experience of running the GISAID platform. And so the Chinese CDC and the labs there chose GISAID to share their sequences, and the WHO was also happy and supporting the sharing of the sequences. This is not a trivial computational feat either, because in the beginning it was a handful of sequences, and when those sequences come in, they come from different sequencing technologies, and sometimes they come with lower quality. So what we did here in Singapore was, initially, we were helping through bioinformatics analysis, aligning to references, comparing differences, to make sure that working with the submitters, that only high quality sequences are coming out. And but soon you see the scale, it became too much to do this manually. So we developed a tool, this is also what Ken mentioned, the Prof server, that does several things. So first of all the genome sequences that are being sent and shared through GISAID first go through a quality check within Prof server. So we are checking if there is a good coverage in different regions, if there is no frame shift mutation. And this is all prepared in a way that our dedicated curators that are spread around the globe, and of course we also have a team here in Singapore, and can look at the quality result and either contact the submitter for improvement or release them to the public through the GISAID website. And you can see, so this plot shows 212. Today we have 261,000 actually already. And you can see also how many countries in the world have managed to submit the sequences. So the first thing when you get a new genome and from a new virus, you can do several things. You want to know how you can detect it. And ASTA has been very early, well, the earliest here, to develop kits to bring it to a broader community, to broader usage. And the excellent colleagues, Masa, Tim, and Sydney, are all crucial to make this happen. And the many people behind them that have helped in their labs and the whole infrastructure. This is something that didn't help just Singapore, but also was helping outside of Singapore. And we are all very proud that this has been a good success story. It is also very important to continuously monitor the sequences. And this is why the GISET initiative is so important. From all globally seen genomes, sometimes we see there are some mutations also in primary regions. For example, here from the US CDC, there was a jump in cases in Korea that had a mutation in one of the primary regions. So just like with vaccines, one needs to continuously monitor the virus. I will come back to how monitoring the virus and interpreting mutation is important. The next point is how can you treat it? Also, there was a lot of mentioning of AI in the previous talk. AI can also help in some things here, and there were some proposals for repurposing of drugs. Some of them, you don't need AI, they were pretty trivial, but at the same time, many of those that were proposed, both by traditional methods as well as AI, didn't really hold up to the promise in terms of making a big difference for the treatment. At the same time, when you have the first genome is also when you can start comparing if antibody cocktails work as drug treatment, as well as start vaccine candidates. And the Pfizer-BioNTech vaccine that was just published a few days ago in the England Journal of Medicine, and also got the emergency authorization that is being now distributed for vaccination in the US, was nicely referencing that their project was possible because of the release by the China CDC via GISET of the genomes. Because it's a regulative framework where every party involved has clarity on intellectual property, as well as the importance of the rapid sharing for the public health is in the foreground. And it's also that when we have the genome sequence that we can ask questions like where did it come from? And that's, of course, a very, very interesting one. And while we were working with GISET and new sequences came in from Chinese colleagues, some of the most interesting ones were, for example, this one was from Zheng Lixi, and this was from a bat in Yuan in 2013. And for a long time, this was the closest precursor to the current virus. So this is a phylogenetic tree where we compare the genomes. So the closer they are in that tree, the closer they are genetically, and they may share a common ancestry. So this light blue ones here is the current outbreak. And then you can see, for reference, down here is the 2003 SARS virus. And here are bat viruses, here pangolin viruses. And you see they all share some ancestry with the current virus. And if you put it together in a map, these pangolins were found in southern China in 2017 to 2019, the closer ones to this virus in 2019. And these bats here, one was in 2013, but also another one here was found in 2019. And so they share the ancestry with the current outbreak. And at some point, the virus jumped from the animals into human. And how does this work? So if we take, for example, that bat virus and we look at the spike glycoprotein, this is on top of the virus where the host receptor interacts, the human host receptor here in blue is bound by the spike glycoprotein. And we just mapped the mutations between the bat Yunnan cave virus versus the current outbreak virus. And you see they're all localized around the receptor binding site here. So the mechanism, the evolutionary natural mechanism for viruses to jump host is sometimes it's a recombination event, but it all requires changes so that the key and lock to enter the new host cell is fitting. And this is what has happened here. So that you can see there is a nice pattern of natural adaptation to the human host receptor. And having seen this, we want to interpret viruses in general. in terms of when they evolve, they will have mutations. Sometimes these mutations can have consequences. Like I showed the one for the host receptor binding, and this is a very typical example. So if you follow from the RNA genome of a virus, you have a mutation here that can affect the mutation at the amino acid level, for example, which can affect the structure level. And if that structure is on that part, for example, that recognizes the host receptor, the single mutation here can affect if a virus can infect, for example, more the upper respiratory tract or more the lower respiratory tract, because there may be different host cell receptors. And this is the case for difference between avian influenza and swine flu, for example. If a virus can infect deep down here, it may be more severe, whereas if it can infect here, it may be spreading faster. And a single mutation in some of the viruses in influenza can help with that. And of course, these two balances may be for different viruses differently, but there seems to be the trend that viruses that infect upper respiratory tract and not the lower respiratory tract tend to spread faster, but are less severe. So that is at least some good news. But how do we interpret this, taking all this into account? We need to know that one single mutation, a viral genome, for example, for coronavirus is 30,000 bases long. So if we have 30,000 bases and we just change one single letter, then it's really hardly any change. So we like to compare it. You have a virus is a car in a certain color, certain type. And if you have a mutation, you're not changing the car type, you're not changing the color, you change a little thing, like for example, a part of the license plate. And we're using the license plate also as an analogy because the license plate in a car tells us where did the car come from, where was it registered. And in this case, it also tells us where did the virus come from. I will show examples later. At the same time, very rarely, and if you compare the frequency, very rarely, you have also another change that is again happening on the outside. It looks like a little change. For example, you can compare it with the volume of the engine of the car. And from the outside, it looks again just like a single little difference. But in the inside, the mechanism, the car actually will go faster with this. And what is the balance? How do we know which one is a license plate mutation and which one is the engine size mutation? And so far, no mutation in this virus has been found that is linked to increased severity. There have been several reports about increased infectivity, for example, in particular a spike mutation D614G. And it's something that naturally happens for the virus. And it's the accumulated evidence put together is that in cells, in some experiments, infectivity is higher. Also in some experiments like hamster transmission, our colleagues have seen increased transmissibility. But so far, all of the clinical data, including data from colleagues in NCID in Singapore, they are finding that although this mutation may do something for the virus, help it spread, but there is no correlation with clinical severity. So this is in agreement with what we knew before. And you can think of it that way. It's something that does happen to the virus and to improve its fitness, but also if it can spread faster without being more severe, it may be more successful afterwards. And how do we computationally help interpreting these mutations? So we developed this tool Cough server that I already mentioned, also helps for the annotation of quality of the genomes in GISAID. And this is also implemented directly in GISAID. So you can look at the sequences and then you can see where is the mutation in the structure. So this is the spike glycoprotein. You see the D614G mutation is further away from the host receptor, which is shown in green here. But instead it's part of a complex. This is an oligomer. That means there are 3 units coming together. And this mutation is in the interface of these units. And this is what we can see here in our tool. And because it is at the interface and it's a strong change from a charge to a smaller residue, it can change the stability of the complex. It can change the openness of the complex. And there are new structural papers coming out in the journal Science that show that with and without the mutation, the stability and the openness of that complex can be changed. And this may be the molecular mechanism, how this mutation does change a little bit for the virus. But again, these changes doesn't mean that it's more severe. And also you need to link the mechanism, the structural part, with the epidemiology. For example, in a phylogenetic tree here, you can see that everything that is yellow here are viruses that have the G mutation, and all the other ones are here. So you see there was an event when this was introduced, and this happened from several Chinese cities, then into Europe. And this was the virus that caused a big outbreak in Europe and then in the US going further. And this is a combination of information that you need to interpret how this mutation maybe is important for both severity, transmissibility, but also later for vaccines. So I already showed you some example about some phylogenetic trees. So this is the other aspect that is very important where bioinformatics can help. And if you think about supercomputing, actually in all these different steps, you could apply it. If you do large-scale genome sequencing, there you have a high computational need there. For structural modeling, if you do very detailed molecular dynamic simulation, you would need a high-performance computing system. And the equivalent for phylogenetic analysis would be Bayesian phylogenetics, for which you need very big systems if you want to do it on scale. So when we compare viruses, we know in this 30,000 base genome, there will always be some mutations. Sometimes these mutations are shared and sometimes they are random noise. So it is interesting that in the very beginning of the outbreak, there was a group of genomes from Wuhan, as well as some other cities, including the first travelers from Wuhan city to Bangkok that were detected in early January, that had identical genomes. So over the whole 30,000 bases, their genomes were identical. And this is interesting because it shows that first of all, there is some conservancy and one can detect and treat using the same methods. At the same time, it also complicates contact tracing. And I will show you another example of identical genomes later on. And it is interesting to follow how the virus evolves. And in the very beginning, it starts in a dense regional area. It's still highly identical. Once it spreads out in different parts in the world, it's like when you create pockets of new evolution. So they will have those license plate mutations, like maybe stamps on a postcard as well. And with these markers, you can tell, for example, that, oh, this virus was seen in Brazil, and there was a traveler that came from Scotland, or this one was from England. And these are these license plate markers that I was talking about. And if you classify these markers into major groups, we call them clades. We classify them based on the early phase. This green one is the one that was found in southern China besides the Wuhan outbreak. The light gray one was the original Wuhan outbreak. So of course you see where the pandemic started. These two viruses coexisted in the beginning. And then you see these orange ones starting to take over everywhere. And these are the D614G mutations viruses, but including with some additional markers. And you see there is the difference between Europe and North America. So they had D614G type viruses, but with some additional other marker mutations. And then we gave them the names GH versus GR. You can also see here, this big peak in Oceania is the one that caused the big Melbourne outbreak. This is a GR virus. So if you're looking at these markers, these genetic markers like license plates, and you have, I'm using here a country, Sierra Leone, which has only 11 genome sequences in the early phase submitted, and you look in the tree compared to the other nearest neighbors, where do they fall into? you can tell that out of these 11 genomes, 7 were independent imports because they are found in different parts of the tree. Sometimes they're from different clades, sometimes they're from the same clade, but in very different parts of the tree that are still distinguished with some additional mutations. And then there were some that were actually very closely clustered together, and these are the sign of some local outbreak. This is when the import happened and then continued spreading within the country. And this is what is also commonly being used. You want to avoid the spread after an import, and the genomics can tell you if you're successful in curbing that spread. And also it can partially tell you where it came from, but that's not always so true. In this case, it looked like that the Sierra Leone cases are often linked to Saudi Arabia. So since we're here in Singapore, I think you will be interested also in how does the global situation compare to the Singapore situation. This is the updated graph from the clades that I showed to you here. Again, everything that is here from the orange onwards is D614G mutation viruses, and the different colors within that are different additional mutations on top of that. So you see the virus keeps evolving as expected. And there is another mutation here, 8222B, that caused this GV virus. Here you can see this peak. This is actually the second wave that happened, spread maybe from Spain to the rest of Europe, mostly by young people. So there was an age bias of between 20 to 26 years old after the summer that had mostly this type of virus, and that were subsequently found in many of the European countries, especially in the UK that does a lot of sequencing. That's why this actually may be a bit overestimated compared to the total prevalence. So you can see that evolution here where the GV strain is here. So now if we look at Singapore, for example, I can explain all of the little peaks here. For example, in the beginning, there was were the travelers from Wuhan. These are the light gray ones. At the same time, there was also some import of this green southern Chinese variant that were co-circulated at the same time, which may even have been in other parts of the world earlier. And but the big Wuhan outbreak was this light gray one. And then mutations from that are followed here. And you see some additional orange peaks here. So the first D614G mutants that reached Singapore were from returning students and returning travelers in late March, when the outbreak became clearer and also people started to move back to their home countries. Now this dark gray one here, this is actually the genomes from the dorm outbreak. And you can see there was an initial rise, but then the control measures fully came into place. And this is a classical example of flattening, successful flattening of the curve. So instead of this going anywhere up here, it was curved and slowly declined. There were also some little flare-ups because it's a very, very difficult scenario to control. But the good thing is we know from this virus decline that the outbreak has been contained, and that it also hasn't resulted in a consistent spillover that would continue circulating in the community. So the virus literally went down to 0 cases from that same lineage. And what you see again, on the other hand, is as people in the world are moving around a bit more, you again get the imports from these viruses, like some GV, some GR, and some GH more recently. And this is something, again, where you need to look. You can capture that virus already with a stay home notice when they come in. And but at the same time, you want to have as few as possible community cases. But if you have them, you can try to link back to which import or which region in the world did it come from. Then you can follow up how did it happen and improve the measures for that. And so far, I think Singapore has been really very good in containing this. And it's a very difficult task, and most other countries are not managing that. So you see all these cases, and Singapore is doing a great job in this. And if you look in detail at some of these examples for contact tracing, this was one of the early clusters, one of 3 early clusters where Chinese travelers were brought to a traditional Chinese medicine shop. You may remember the story with the tour guide and then also a staff working there. And that brought the virus back home, where it infected the husband, the son, and the domestic helper. And if you compared the viral genomes from these cases, you can see that the 3 cases at home, so very close transmission context, are also again identical. So over 30,000 bases, they are identical. At the same time, they are 1 mutation away from the one who also got infected by the Chinese travelers. And they are 2 mutations away from the original outbreak in Wuhan. And the genome tells you this. And I also have to say that it's not a solution for all kinds of contact tracing, because there is some ambiguity. There may be other genomes that are similarly close phylogenetically to some of the cases. And then you can't decide, did it come from this country or that country? Did it come from this chain or not? However, sometimes there are specific characteristic markers, for example, deletions or a set of unique mutations that allow you to classify things into transmission chains. And one such mutation has happened, a large deletion that the colleagues in Duke NUS have found, and we also clinically described the colleagues in NCID and and ASTA. And this is a huge deletion with 382 base pairs that actually attenuated the virus. And this was found in 3 different clusters here. It was found only once, otherwise in Taiwan. It is likely to have originated in China, brought in by the travelers. But this, because of the uniqueness of this deletion, once it was brought into Singapore and caused some clusters of maybe 30, 40 people each, through the genetics, we could see that they were linked, but also through the genetics of the continuous surveillance afterwards, we could see that the countermeasures were working and that this virus got extinct. So this is, again, an example where you can get confirmation from the genomic surveillance if a virus has stopped circulating and if that cluster with a unique marker you have contained. At the same time, a similar thing happened. There was a conference in a hotel in Singapore where a lot of people from different countries came, and this still happened very early in the outbreak, so that the conference was actually still happening. And one of the cases was the 3rd UK case, and he traveled further to Switzerland in a nice chalet in France, and along the way people got infected. And we know again that this is an actual transmission chain because there is a deletion involved. This is a much smaller deletion, unique 3 base pair deletion. And while we don't see this deletion in Singapore, so although it may have passed through Singapore through that conference, we do not see cases with that deletion in Singapore. So again, the control measures have worked. However, we see thousands of cases in Europe with that deletion as a follow-up from the further trips. And what is not listed here, it also was found in Spain. And often it's people traveling for holidays, etc. I want to give one other example that is related to international imports. So the outbreak may have started in China, in southern China, maybe at the same time in Wuhan city, where the biggest outbreak was recognized. But at the same time, the virus afterwards was actually quite well controlled in China. And there was sequencing in China. And when the sequences from China were reported, we could see that these are not anymore the ones directly from the outbreak that they have stayed there, but actually they have gotten those new license plate mutations that we know that they actually may have come back from Europe. And it's also interesting in this particular case, this was in October in Qingdao, where 2 workers handling frozen food were infected. And then colleagues from China CDC also found on the packaging viral RNA, and that was not just PCR positive viral RNA, but it actually could show that this was RNA that can still infect cells. So it was infectious virus, and the virus from the packages was closely matched to the dog workers that were handling it. So that is one of the possible routes through the cold chain that the virus can spread. And one needs to, of course, continue monitoring all ways of spreading from travel, but also trade. So now there was all big news about vaccines starting to be rolled out, and that's really promising. And one question that many people, of course, naturally have. So now, when the virus has mutated, as you have seen with all these little mutations, does this affect how the vaccine would recognize it? And the answer is the vaccines designed from the first version are still very effective. So we know from influenza that sometimes there is antigenic drift and there are some differences. There have been some reported cases of reinfection. That means that your body's immune system either didn't work in the first place or the virus has slightly changed so that your immune response is not effective in neutralizing the slightly modified virus. And in some of these cases, we see there are some patterns that change conformation of glycans, sugar on the surface. And this is something that can change how an antibody can recognize and bind. This is also what happens gradually with influenza, and it may mean that there are escape mechanisms that at some point in the future, one may have to update vaccine strains. And this is why the big organizations, COVAX, CEPI, supported by WHO, are also working with GIS8 to keep monitoring these mutations and in regard also to the antibody binding. And this is a collaboration also. I'm not showing all the data that we have with DSO in Singapore. And they also have been monitoring and doing experiments on in determining if there is change in receptor binding as well as in antibody binding. And I mentioned there was an outbreak in Melbourne that was this big orange spike. What is characteristic for that Melbourne outbreak actually is also that it had a new receptor binding mutation, S477N, that occurred very commonly there. And I will show you also some other complementary data that this also can be one that can change a bit how the virus interacts with the host cell, but it doesn't mean that it's necessarily linked to severity. So our colleagues in Melbourne are also analyzing transmissibility as well as the viral kinetics. And from the viral kinetics, they did not think it was more infectious. And in our tool, the COF server, this is how the mutation looks like. So you see different from the D614G mutation that is here. This one is actually at the receptor binding interface, a bit on the outside. So it depends on the definition of distances, if you include it or not. And our tool can tell where did it occur and when. And also it's linked to literature. And besides the ongoing work with colleagues in Singapore, there are also other measures already available for this mutation that it does slightly increase the binding to the host cell receptor. And also we know that in crystal structures, it's in position where antibodies can bind. So keep in mind, vaccine response may be multiple antibodies, a polyclonal response. And that means one single mutation will not affect that the vaccine doesn't work anymore. But one interesting facet of this mutation is that although it caused a big spike in Melbourne, it is not unique to that genome that was found there. It's actually, this for example, here's the Melbourne outbreak in the tree, the yellow thing, but you see there are also other yellow dots in between here. And this means that this mutation does occur, is not so much a license plate mutation specific for Melbourne, but actually can occur in many different contexts. And currently there is about 50% of strains in the clade GH that occur in Europe with this mutation. And that's an interesting facet. So I want to summarize that real-time surveillance is possible for viruses with modern technology, starting from next-generation sequencing to multiple parts of computational analysis and interpretation, both from phylogenetic as well as structural level. And through that, we are able to detect it, start to treat it, trace it, contact trace it, which is also important for making sure that you know that it's contained. We know a bit where it came from, but there will be, of course, more studies and more knowledge available in the future. And very importantly, we can trace how it changes. This is important for looking for diagnostic primer stability, as well as for the future vaccines. So I will end here, and there are many, many people to thank. I'm just listing explicitly not just our workers in BI and also GIS that have helped, but also young interns that have joined us doing this whole work, helping GISAID and helping the world with many of the great efforts. And in Singapore, it's a whole of government effort, many agencies, many colleagues in ASTA, but also across the sectors. And of course, key stakeholders, MPHL, NCID, MOH. They all help to work together, and we are very happy to work with them. And of course, we're grateful for funding that we received to do this work. Okay, thank you.
All right, thanks a lot, Sebastian. Very interesting stuff about COVID, and I'm very happy that Singapore is playing a large part in actually surveilling its changes globally as well as in Singapore. So right now we have a panel discussion. I know we're running a little bit late of time, but I'm very happy that we do have questions along the way. Thanks, Prof. Ngiam, for answering all the earlier questions. So I'll just jump in and go in order. So we have a question from Ricky here. He's from NTU. I think maybe this one, Prof. Ngiam may take it because he's asking about how do you handle the data security aspect of it? Because I think for GSAID data, that one is more public, right? Prof. Ngiam, would you like to take this question?
Thanks, Kenneth. So we generally don't share healthcare data to the public. So all the healthcare data that are related to hospitals and public healthcare is shared by MOH. So MOH takes the lead in sharing this kind of data on data.sg. So that's the aggregate national level statistical data that one can access. But specific to institutions and to, of course, certainly to patients, there's no way that data is going to be shared. Yeah.
All right, thanks very much. Next question in order is by Amir Ali. So I believe this is more for Sebastian. So he congratulates you on your new position in January, which is coming up very soon. He's asking, how is it different from analyzing H5N1? Is it that much more different analyzing COVID-19?
Yeah, well, thanks, Amir. I also I remember you were at BI, I think, for some time. And so the SARS-CoV genomes, they are mutating a bit less. It's also a non-segmented virus, which makes that part easier. Otherwise, you need to look at the individual segments separately. And H5N1 also, there is currently an outbreak of H5N8 that has gone from Asia into Europe in wild birds. So there is an animal pandemic happening at the very moment, and with H5N8. And luckily, this is a virus that doesn't infect humans easily, but there are mechanisms how these viruses can change. So we also need to look at that closely. So GISAID has been working on avian influenza viruses for a long time. So while we are dealing with the SARS-CoV-2, we are also continuously looking at the avian influenza outbreaks.
Thanks very much. Maybe I open the next question to both panelists. So there's some question in the chat. It's a bit out of order, but I thought that's a common question that could apply to both. So Jordan here is asking about translating domain knowledge, whether it's related to virus sequences or in terms of Professor Ngiam's case, clinical data, into a program for supercomputers. So a lot of them actually use high-performance computers. When you're doing AI, it's a GPU card is by definition a high-performance computing platform already. So it's asking, would this be a job for the scientists alone, or how in your respective institutions you get a domain expert to work with computational people to get things done? Maybe I ask Professor Ngiam first.
Yeah, so right now we are just ensuring that the pipeline in which the sequences are made and can be called and then can be stored in a way that researchers can access it in a relatively rapid manner. Again, sequences themselves have no identifiers, right? The real challenge here is to be able to do downstream linkage and association studies with at the population-wide level. So traditionally, this would be the GWAS things that we do. So we are really trying to solve the problem of true stream kind of sequencing to bedside kind of processes that allow our researchers and our clinicians to be able to use that data effectively.
Thanks very much. So, I think something that I think, if I read the question that Jordan is asking, is that typically a domain expert, so a clinician, or it could be a wet lab scientist, for example, within A-star like that. How would that person actually interface with a data scientist or bioinformatician in your part? Any particular challenges? So maybe Prof. Yuan can just comment on that, the human aspect of that, where the resources, who do we talk to?
Oh, Kenneth, you probably be more familiar about this than I am, but we know all the challenges, which is why it's important to have forums like this, and as well as in the past week where we had Kenneth was actually teaching at the executive education course for healthcare AI. These forums allow us to interact with the clinicians and the data scientists in a way that you won't normally find them in the same room. I mean, how often do we find a clinician, a data scientist, and a basic scientist in the same room, right? Let alone in the same Zoom room, right? So I would say that we need to create occasions for people to meet. Whether or not there are formalized structures for people to come together largely depends on which is the initiating organization. So in this case, I would imagine that a healthcare sector cluster like NUHS would be a good initiating organization to initiate these conversations and collaborations between scientists, data scientists, and clinicians. Because in the end, right, we need to be able to deliver the insights and the care to our patients, right? And also the source of a lot of these material comes from patients. Yeah.
Thanks very much. So, Sebastian, you're the heart of the BII. You have been working with NCID, NPHL, many other acronyms of things like that. How was it for you that you could help to translate the ideas of the domain experts to bioinformatics and high performance computing?
So I think it is at the time of a pandemic, when you need to move fast, domain knowledge becomes even more important because it speeds up the process. And I would say that the likely scenario that we often encounter is that we have computer scientists, we have, let's say, the clinician, the domain knowledge, and we need still someone in between, a biomedical informatics or bioinformatician specialist that helps translating between the two. And an example where this is necessary is if you have the algorithms for sequence assembly, for example, the virologist will know a mutation. This is not likely to be real, or that looks like noise, or there are too many holes in the sequence. The computer scientist wouldn't know that this is a problem. And the bioinformatician in the middle can say, oh yeah, this is a problem. It comes because the way the algorithm stitches this together, you need to open that gap a little bit, and then it fits in. So this is a real world scenario where you can move faster if you have a good combination and collaboration between the computer scientist, also data scientists, the AI people, with the domain expert. And the domain expert is a bioinformatician that knows some programming and that knows some biology or virology. And they translate then to the stakeholder who is the clinician. And this can be a very fruitful combination. And this is also why in our institute, we have a mix of people that cover this. And in different projects, this can be very important to move fast.
Thanks very much. So from NSCC angle, of course, we hope to enable any platform that wants to be scalable. And of course, hopefully this forum is just one of many forums. We have forums in each of your own institutions, but at NSCC, hopefully we bring it at a national level. So I think maybe time for only one question, but unfortunately, I think there are 3 questions here. I think directed at Sebastian. From Tin Wee, our Chief Executive at NSCC. So you can see there are 3 questions down there. So I think in the interest of time, maybe you can pick the one that you want to answer most, that's most germane, and then the rest of it, maybe we can take it offline also, because I believe everybody is busy and may have to move off soon.
I'll answer it quickly. So I talked to Tin Wee, of course, and he was one of the first that I showed at a precision medicine meeting some of the phylogenetic trees that we were making. And I think this is a good example already that we are somehow prepared for disease X, because it was, many people may have looked for influenza, but we have seen a new coronavirus, which is also on the list. And mechanisms were in place to both create diagnostics quickly, as well as do adapt the computational tools. This is what we are responsible for. And together with all our colleagues in Singapore, whenever there is a need, we are a well-established community. So we know that if we need a high-performance power, we can go to NSCC. And I think having systems in place, practicing on the new virus like this coronavirus, as well as in peacetime, continue on circulating virus like influenza, or new viruses, not new viruses, but viruses that are new under surveillance and that also fall into WHO networks, is RSV, for example. And the more we are able to work with each other from NCD via BI to NSCC, we have good examples to be prepared for the new diseases.
Thanks very much. So we thank the 2 panelists, but I can see a possible convergence here. From, for example, a healthcare institution, I mean, if the data not just flows to NSCC, but actually surveillance nationally for anything that could actually have forward projections, that could be a mutation that could be more infectious, or having more clinical severity, and it feeds down to the real-time data that Professor Ngiam was talking about. That could actually influence, clinical decision making, even changing the logistics of how healthcare manpower is. So, I think this is a great time. I'm very happy that the 2 panelists here. There are many things of interest, healthcare that affects everybody. Are there any other things I'd like to bring up? Maybe I think Professor Ngiam, would you like to any last words before we sign off?
Yeah, I think I want to address one of the points about advanced early warning things. One of the things that we did many things well in Singapore. I think we have to give credit where it's due. But one of the things that we didn't do very well is to ensure that the data is shared nationally, right? I think this will have, I believe that this will have changed the trajectory of the COVID spread in Singapore. And it all comes down to the willingness of multi-agencies to share this data. And I've always expressed this, and Tin Wee and Kenneth, you know that I think NSCC is the right spot to share that information. So yeah, I hope you can convince the powers to be.
We need people like you and Sebastian yourself to also have a united front to convince the powers to be. That data sharing responsibly is the way to go. Sebastian, any last words before we sign off from this very excellent 2 talks by
I enjoyed the talk. So data sharing always is a problem, also internationally. GISAID is one step to improve this. And Singapore has been contributing to that. So 1,500 genomes from Singapore are there, and also the other ASEAN countries are contributing too, which is also rare event. And I wanted to say that for precision medicine, there is one area that we don't understand so well. Why does it cause more severe disease in some people, not in others? And it's most likely not changes in the virus, but in the patient. So exactly your line of work with precision medicine and also using AI to analyzing some of the responses, like the immunological responses, will lead to improvements in understanding when the virus is a problem for a patient or not. Thank you.
All right, thanks very much. So I think we have each other's contacts, and I encourage everybody, the audience also, to contact any of our two panelists as well as NSCC for possible collaborations. There are some questions about the slides. I believe Dr. Sebastian's slides can be shared. Prof. Ngiam has some proprietary information, so unfortunately that cannot be shared. All right, so but do keep in touch with everybody, and then hopefully we'll keep this momentum going. And thanks very much for the. So hand it back to Alethea.
Thank you, Prof. Ngiam, Dr. Morristra, and Dr. Ban for the great and insightful sharing, and thank you to everyone for the ongoing questions. Both speakers have shared their email addresses, so feel free to get connected with them after this session. The recording of this session will be uploaded on NSCC's YouTube channel, so do head there for a review of today's event. So that concludes today's session on pandemic-proofing society. Do keep an eye out for the next series of webinars that we are planning for in the new year. Details will be posted on our website and social media channels. So see you at our next online event. Bye.
Bye.
Okay. Thanks everybody, and stay safe.